Welcome to High Body. High Body aims to introduce the fundamental anatomy structures and biological functions of the interconnected organ and tissue systems of the human body. In this episode, let's start our journey of discovering the mysteries of human cells and their organelles together with High Body. Human cells are the basic structural and functional units of life, serving as the building blocks that construct our bodies and enable them to function. It is estimated that the human body contains thousands of different types of cells with total around 30 trillion cells, each tailored to perform specific functions within the body. Below is a small selection of human cell types. Stem cells are undifferentiated cells capable of self-renewal and differentiation into specialized cell types. Muscle cells, also called myocytes, are important for a range of functions, including movement, support, and internal functions. Bone cells have at least three main types: osteoblasts for forming new bone, osteoclasts for dissolving bone, osteocytes for communicating with other bone cells. Blood cells have many types: red blood cells for carrying oxygen around the body, white blood cells, neutrophils, and basophils as part of the immune system, platelets for helping blood clot to prevent blood loss after injury. Fat cells, also called adipocytes, are the main adipose tissue constituents. Nerve cells, also called neurons, form the communication system of the body. Sperm cells with tadpole shape are the smallest cells in the human body. Female egg cell is the largest human cell. All human cells contain cell membrane, cytoplasm, and nucleus except the mature red blood cells. Cells are surrounded by cell membrane, also called plasma membrane, which forms an important barrier or boundary between the internal and the external cell. And cell membrane is a phospholipid bilayer composed of amphiphilic lipid molecules, phospholipid and cholesterol, and interspersed or embedded membrane proteins. The polar heads of lipids are arranged on both the inner and outer surfaces of the cell membrane. The nonpolar tails of the lipid layers face each other in the center of the membrane. Cell membrane is selectively permeable to regulate what moves in and out of the cell. It is permeable to such molecules as oxygen, carbon dioxide, water, steroids, and other lipid-soluble chemicals. Other substances, such as glucose, ions, or proteins, cannot pass through the cell membrane and cross it only by specific transport mechanisms. Cell membrane has the functions to maintain cell integrity and electric potential while allowing selective permeability. Cell membrane also contains a range of receptors that are crucial for intercellular and intracellular communication and nutrient uptake. Internal to cell membrane is the cytoplasm, which includes the jelly-like fluid and the membrane and non-membrane bound organelles. The jelly-like fluid consists of water, proteins, ions, and other molecules necessary for cellular metabolism and transport, which provides a medium for cellular activities to occur. Numerous organelles with complex network are also embedded in the cytoplasm. Each organelle has specific structures and functions to perform a specialized metabolic function that is essential for cell life. In most cells, the nucleus is also located within the cytoplasm. The cytoskeleton of a cell consists of a continuous network of tiny protein filaments and tubules that extend throughout the cytoplasm, from the nucleus to the cell membrane. Mainly three types of filamentous proteins form the cytoskeleton of a cell, which includes microfilaments, intermediate filaments, and microtubules. Cytoskeleton serves as cell's structural framework to maintain the correct shape and mechanical resistance of a cell. However, unlike regular scaffolding, the cell cytoskeleton is flexible, and it plays a role in cell division and cell motility. Nucleus, often referred to as the control center of the cell, is the largest organelle of a cell. Most cells have a single nucleus, but other cells may exhibit multiple nuclei. Skeletal muscle cells have multiple nuclei, whereas mature red blood cells of mammals do not have a nucleus. A double membrane called the nuclear envelope separates nucleus from the rest of the cell. The outer nuclear membrane is studded with ribosomes and is continuous with the rough endoplasmic reticulum. The nucleus also consists of nuclear lamina, gel-like nucleoplasm, chromatin, one or more nucleoli, and nuclear matrix. And it is the storehouse of the cell's genetic material DNA deoxyribonucleic acid in the form of chromosomes. At intervals around the periphery of the nucleus, the outer and inner membranes of the nuclear envelope fuse to form numerous nuclear pores. These pores function in controlling the movement of metabolites, macromolecules, and ribosomal subunits between the nucleus and cytoplasm. The functions of nucleus include regulating gene expression and controlling cellular activities by transcribing DNA into messenger RNA that serves as a template for protein synthesis. Additionally, the nucleus plays a crucial role in cell division, orchestrating cellular processes such as mitosis and meiosis. Endoplasmic reticulum is a complex interconnected network of membrane-bound tubules and sacs extending throughout the cytoplasm. There are numerous of endoplasmic reticulum in a cell categorized as two types: rough endoplasmic reticulum that is covered or studded with ribosomes, and smooth endoplasmic reticulum that is lacking ribosomes. Rough endoplasmic reticulum is involved in protein synthesis, where ribosomes translate mRNA into proteins that are then folded and modified within the endoplasmic reticulum lumen. Smooth endoplasmic reticulum, on the other hand, participates in lipid synthesis, detoxification of drugs and toxins, and storage calcium ion. Ribosomes are non-membrane-bound organelles that contain ribosomal RNA and ribosomal proteins. A typical human cell may contain thousands to millions of ribosomes. Some ribosomes float freely in cytoplasm while others attach to endoplasmic reticulum. Ribosomes are the cellular factories responsible for protein synthesis. Ribosomes read the genetic code carried by mRNA and assemble amino acids into polypeptide chains according to the sequence dictated by the mRNA. These newly synthesized proteins undergo folding and processing before being transported to their designated locations within or outside the cell. Golgi apparatus, also called Golgi complex, consists of flattened membrane-bound sacs called cisternae. A typical human cell may contain a few to dozens of Golgi apparatuses. Golgi apparatus are the cellular post offices that are responsible for receiving, processing, and packaging proteins and lipids synthesized in the endoplasmic reticulum for distribution to various cellular destinations. Mitochondria consist of an outer and inner membrane. The inner membrane exhibits numerous folds called cristae. A typical human cell may contain hundreds to thousands of mitochondria. Mitochondria, often referred to as the powerhouse of the cell, are responsible for generating ATP adenosine triphosphate, the cell's primary source of energy. These double membrane-bound organelles contain their own DNA and unique ribosomes, allowing them to partially self-replicate. Mitochondria convert nutrients such as glucose and fatty acids into ATP through a series of biochemical reactions known as cellular respiration to provide the energy required for cellular processes. Lysosomes are membrane-bound organelles produced by the Golgi apparatus. A typical human cell may contain dozens to hundreds of lysosomes. Lysosomes contain digestive enzymes that are responsible for digesting and removing wastes, foreign particles, and dead damaged cells through a process called phagocytosis. These enzymes function optimally in an acidic environment maintained within the lysosome, which allows for efficient degradation of macromolecules into smaller and recyclable components that can be reused by the cell. Peroxisomes are membrane-bound organelles that appear similar to lysosomes, but are smaller. They are found in nearly all cell types. A typical human cell may contain dozens to hundreds of peroxisomes. Peroxisomes contain enzymes involved in various metabolic reactions, including the breakdown of fatty acids and detoxification of harmful substances such as hydrogen peroxide. The centrosome is an area of the cytoplasm located near the nucleus. There are two mutually perpendicular small cylindrical structures known as centrioles and the surrounding matrix within the centrosome. Each centriole consists of 9 evenly spaced clusters of 3 proteinaceous microtubules arranged in a circle. The microtubules have longitudinal orientation and are parallel to each other. Most human cells have one centrosome. Centrosomes play a crucial role in organizing the microtubule and cell division. The centrioles in the centrosome replicate and form 2 pairs before mitosis. During mitosis, each pair moves to the opposite poles of the cell, where they become microtubule organizing centers for mitotic spindles that control the distribution of chromosomes to the daughter cells. In summary, human cells and their organelles work harmoniously to sustain life. From the nucleus's genetic instructions to the mitochondria's energy production, each component contributes to the cell's overall functions and vitality. Thank you for your interest in HiBody. Let's continue our journey of discovering the mysteries of the human body systems together. And stay tuned for more episodes from HiBody.